conducted your research at?
I work in a research lab at the University of Michigan's Kellogg Eye Center. It's the same university I did my undergrad in, and I started working right after.
Great. Tell us about the poster that you presented here at ARVO 2017.
This poster was about programming in ImageJ and Fiji. We have so many images from all our experiments that were waiting to be analyzed, and they're about subcellular structure counts moving up and down when you do assays. So things like lipid droplets that we label with anti-ADRP, or autophagic vacuoles that we label with anti-LC3, or lipofuscin that we build up in our cells. All of this is in a human fetal RPE model. We end up with these immunohistochemistry and immunocytochemistry data, and we need to count all of these tiny particles. And you could get somebody to do it manually, but it's just faster, more efficient, and less inconsistent when you get a program to do it. But getting a program to recognize what the structures are is the challenge. So you have to make it think like the researcher wants it to think and recognize the structures. So that's what the poster was about.
So what were the results of your study?
Well, the results initially were that this may not be entirely possible. It may be just about as accurate as manually counting it, because even the program would have some sort of rate of error, because the images you provide may not be the same from experiment to experiment, and you can teach it with one data set, try it on another data set, and tweak it again, and then tweak it again. But maybe it wouldn't be able to encompass everything you could throw at it. But we worked on it for a really, really long time, and we gave it every shade of data set we could find over a period of maybe more than a year. And so now it works so accurately that we no longer also manually count to fact check our program data. So we trust it now.
What do you think the potential clinical implications of your work might be?
So the structures that we made this program for, they're all involved in studying the pathogenesis of AMD. So macular degeneration affects such a huge segment of the population, and especially the aging population. Everyone's trying to figure out how they would make therapeutic options available, either preventive or for treatment. Just after it's begun to develop, it just progresses so fast. And there are anti-VEGF therapies for wet AMD, but dry AMD is just so hard to treat. So these structures that we're tracking, we can then use the analysis of these to test drugs. For example, another interesting project that I'm working on right now is autophagy induction. It's how you could get the RPE to start cleaning up the material that would otherwise build up and cause macular degeneration. So if we can use tools like these to help out other research areas, that could transfer into getting drugs to clinic.
I think it's a very fascinating line of research that has enormous potential. Where do you see yourself with this project a year from now and 5 years from now?
This project, it depends. I learned an entirely new coding language to be able to help the lab out with all these images. And it's such a vital skill set. Image analysis, no matter what field of science you're in, you'll need it. It's like statistics, right? So basically anything that needs to be done, any sort of analysis, this software will adapt for. So you can write new code for it, or you could use the existing plugins. ImageJ is all about tools that other scientists put together, and so it's very collaborative, and there's a lot of back and forth. We have email listservs, and so we can update each other every day about what our problems are, what our solutions are. So I see myself using this for the rest of my research career, yeah.
Tell us about some of the other projects that you're working on in this lab.
Yeah, so like I mentioned, we're trying autophagy induction. We test inducers and inhibitors to see if we can get it to start clearing up material like lipofuscin. And so we work on human fetal models, and so they don't come with the lifetime of lipofuscin that we would see in an adult human eye. But we build it up in artificial model in vitro, and we try to test drugs while we're building it up to see if it would prevent the buildup, and also after it's been built up. And we're getting some interesting data, which we haven't published yet, but it would be really exciting. We're testing FDA-approved drugs that have been approved for other diseases, and also drugs from other countries that have been approved there, again, for other treatment options. But it looks promising.
What agents are you looking at?
So everybody associates autophagy with mTOR regulation, and some of our drugs are related to the mTOR pathway, but some of them are proteasome related or lipid metabolism related, and so there are a lot of pathways that if you tweak, you could alter the way the RPE handles oxidative stress or photoreceptor outer segments that it's ingesting and how it chooses to handle the material.
So, in summary then, let's put this all into context. Tell us in 1 or 2 paragraphs what you believe the ultimate clinical outcome should be from your research.
So I guess that would be having some sort of really easily integrated dietary supplement that you could give somebody from a certain age up to just promote eye health to the point where AMD wouldn't be such a concern for everybody over the age of 60. People start to lose their vision and they don't realize it. You could foreseeably lose the entire vision in one eye and still not realize it. But if you're on a regimen of, say, I mean, everybody takes vitamins now, maybe more than they should. But if you could say, okay, from this age onward, take a baby aspirin. It's just what we know works. If we could do something like that for retinal health, then it would really be the best solution.
Right. I mean, that raises the issue of polypharmacy.
Yeah.
As you know, people take herbal remedies, vitamins,
Yeah.
sometimes mega amounts of vitamins.
Yeah.
It obviously changes the milieu and it changes the challenges. So what you're saying is that people have to treat vitamins as medications.
Well, it wouldn't be like a vitamin. It would just be something that you would take as preventive.
Right.
Yeah.
Excellent. Is there anything else you'd like to share with us?
There's a lot more posters, presentations, papers coming out of Kellogg, and we'll be here for the rest of our and all of our research is really amazing.
Thank you for your incredible work, and we look forward to watching your progress and learning from the results.
Thank you.
Thank you.